Hello, everybody, and welcome to this discussion on plasmids. So plasmids are extra chromosomal replicons, and in nature, they confer various phenotypes. And plasmids whereby the phenotypic traits have not been elucidated are termed cryptic plasmids. Now, plasmids can be categorized into various categories. You have conjugative plasmids. These tend to be of a higher molecular weight, and they tend to engage in stringent replication. Now, conjugative plasmids contain a set of transfer genes termed the TRA genes. On the other hand, there are non-conjugative plasmids. These tend to be of lower molecular weight and tend to be relaxed. They also do not contain the TRA genes required for engagement in conjugation. We can also classify plasmids based on their replication properties or characteristics. We have stringent plasmids and we have relaxed plasmids. Stringent plasmids are maintained as one or a few plasmids per cell. For example, 1 to 3 plasmids per cell. In other words, they have low copy numbers. On the other hand, relaxed plasmids are maintained as multiple copies per cells. In other words, they have a higher copy number. Now, we said that plasmids are extra chromosomal replicons. Now, they are double-stranded and they are also circular, albeit there are exceptions for the circular aspects. So, linear plasmids exist in some bacteria, with ends being protected by either hairpin loops or covalent protein attachments. What you have to recognize here is that in double-stranded plasmids, if both strands are intact circles, these are termed covalently closed circles or cccDNA. And if one strand is an intact circle, then this would be an open circle or OCDNA. Now, OCDNA and cccDNA or covalently closed circles DNA can switch from one to the other. So DNA ligase can act upon open circles DNA and make it such that it is covalently closed circles DNA. And cccDNA can be acted upon by endonuclease to make it OCDNA. Now, we can separate cccDNA from OCDNA by gel electrophoresis in agarose gel. Also, what you have to recognize in terms of covalently closed circles DNA or cccDNA is that when isolated from cells, it contains deficiency of turns in their double helix. They are supercoiled. Now, we said that cccDNA and OCDNA can be separated from one another by gel electrophoresis in agarose gel. Now, this is whereby supercoiled DNA travels a greater extent down the gel than OCDNA. What you have to recognize is that we can visualize this separation by using ethidium bromide as an intercalating agent, keeping in mind that an increase in ethidium bromide will result in increased untwisting of the double helix until supercoiling occurs in the opposite direction. Now, I mentioned that plasmids can be categorized into various categories, and I mentioned conjugative plasmids as well as non-conjugative plasmids, and I also mentioned stringent plasmids as well as relaxed plasmids. I also told you that stringent plasmids tend to have a low copy number, and relaxed plasmids have higher copy numbers. This leads us to a discussion of plasmid replication, whereby plasmids only encode a few or only one protein required for their replication, which tends to be in close proximity to the origin of replication. And so in plasmid replication, you have to keep in mind that plasmids only encode a few or only one protein required for their replication, and they tend to be in close proximity to the origin of replication, and thus only a small region surrounding the origin of replication is required for replication. This can be termed the essential region, quote unquote, and in turn, other parts of the plasmids can be removed or sequences can be placed or added in place of them, And so this facilitates the use of plasmids as cloning vectors. Now keep in mind that when we discuss the origin of replication, that this determines the host range of the plasmid. So the origin of replication determines the host range of the plasmid. For instance, an origin of replication derived from plasmid col E1 replicates only in enteric bacteria such as E. coli, and an origin of replications derived from plasmid RP4 replicate only in gram-negative bacteria or most gram-negative bacteria. And then origins of replication derived from plasmid RSF 1010 replicates in a broad host range such as gram-positive and gram-negative bacteria as well. And plasmids with broad host range, you should recognize, tend to encode most, if not all, of proteins for replication. So I told you in reference to plasmid replication that plasmids only encode a few or only one protein required for their replication. And for replication, they are dependent on the replication machinery of the host cell, such as the DNA polymerase of the host cell, DNA ligase of the host cell, and helicase of the host cell. Yet, the copy number of a plasmid is determined by regulating the initiation of plasmid replication, of which two mechanisms have been elucidated: regulation by binding of essential proteins to eterons, and regulation by antisense RNA. Let me discuss regulation by binding of essential proteins to eterons. What you have to keep in mind is that eterons are repeated sequences. So take, for example, the PSC101. The PSC101 vector was the first cloning vector used by Stanley Cohen and Herbert Boyer as the very first cloning vector. Now here, the origin of replication region contains 3 to 7 copies of an eteron sequence that is 17 to 22 base pairs long. In close proximity to the origin of replication is the Rep-A gene. The Rep-A gene encodes the Rep-A protein, and the Rep-A protein binds to the ORI region that contains 3 to 7 copies of an eteron sequence that is 17 to 22 base pairs long. What you have to recognize is that the Rep-A protein binds to eterons, and in so doing, initiates DNA synthesis and thus plasmid replication. So in terms of the PSC101, the copy number is regulated by two interrelated mechanisms, whereby Rep-A protein represses its own synthesis and Rep-A protein can link two plasmids together. So the Rep-A protein represses its own synthesis by binding to its very own promoter, thus blocking transcription. You have to keep this in mind. In addition, the Rep-A protein can link two plasmids by binding eteron sequences of two plasmids, thus preventing replication of the plasmids. So we've discussed the regulation by binding of essential proteins to eterons, which are repeated sequences. Let me discuss regulation by antisense RNA. Now, regulation by antisense RNA is whereby you have RNA1 and RNA2. These are RNA molecules that act to control and regulate replication. Here, you have to recognize that RNA2 ribonucleotide forms an RNA-DNA hybrid at the origin of replication, and in the process primes for plasmid replication, and thus increased levels of RNA2 ensues when the copy number is low. Now, RNA2 must first be cleaved by RNase H to leave a free 3' hydroxyl group so that it can act as a primer for replication. Now, on the other hand, the RNA1 ribonucleotide can form an RNA-RNA hybrid with RNA2, and in turn, this prevents priming for plasmid replication by RNA2, and thus increased levels of RNA1 ensues when the copy number is high. Now, when I say that increased levels of RNA2 ensues when the copy number is low, and increased levels of RNA1 ensues when the copy number is high, this should tell you that there is a detection system whereby we can detect, or the plasmid can detect, when their copy numbers are low and when their copy numbers are high. In reality, when we delve into this can get very complicated, and we don't fully understand plasmids to the extent that we wish to understand them. Now, what you have to recognize is that in addition, there is something called the rope protein. The rope protein binds to dimers of RNA1, RNA2 hybrids, and as a result, enhances RNA1, RNA2 binding. Now, because the rope protein binds to dimers of RNA1, RNA2 hybrids and enhances RNA1 and RNA2 binding, this causes maintenance of the copy number by inhibiting processing of RNA2 even at low concentration. So the rope protein modulates the activity of RNA1. So the rope protein makes it so that the RNA1, RNA2 hybrid, or the dimers of RNA1, RNA2 are enhanced, or their binding is enhanced. And thus, if we delete the rope protein, then this causes an increase in copy number, and this is something that is done. We can delete the rope protein, and in so doing, we increase the copy number of the respective plasmid. In addition, you should recognize that mutations in RNA1 also results in increased copy numbers.